Good afternoon to all of you. Kiran is a professor in the Department of Bioscience and Bioengineering. He's broadly in a broad research areas viruses. He looks at their evolution, ecology, molecular mechanisms, and such other aspects. Kiran is one of the star faculty of our department. He leads a very large and very active research group, and he has made very important contributions. He's also a recipient of Excellence in Teaching Award. He teaches a course on evolution, which is very, very popular among undergraduate students. A large number of students every semester credit that course. Kiran is an entrepreneur. He's a co-founder of a company and which has a headcount of probably about 150 masters and PhD students. And I think that's a very phenomenal thing, and it is growing. And he has also contributed to the Institute for Administration. Quite a few things he has done. One of the things is that he has been in charge of undergraduate admission, JEE seed as well as USEED. And especially among a couple of years which were quite difficult, he led the institute effort towards that. So overall, I see Kiran as a complete package for the institute: entrepreneurship, teaching, research, and administration. So excellent, Kiran. One last thing before I hand over the stage to you is that, as far as I remember, this is the third or fourth time he's giving lecture on Nobel Prize. It just tells that the kind of expertise he has and what kind of various Nobel Prizes are given, there seems to be some kind of a match. Excellent, Kiran. Okay, over to you.
So that's very flattering, but I can top that. I just got a message from my daughter. Maid just walked in and she congratulated me for the Nobel Prize. So she thought that IIT Bombay this year got 3 Nobel Prizes and I got one of them. So maybe we should see how we can put up our posters. Maybe not put our photos, maybe include, add actual winners photos. Anyway, so today I'll tell you a little bit about why this mRNA vaccine got the Nobel Prize. And these two scientists, they made actually a very small tweak to the protocol of making this vaccine. So, but before this, I'll give you some context to their discovery. I think most of you are familiar with how vaccines work. And I'm pretty sure you're also familiar with this term antigen, right? It is some kind of a foreign substance that induces an immune response. And most of the viruses are like foreign substances, virus, bacteria, or any of the proteins or any material that is coming from some external environment, right? So they actually, when they get into our body, they induce a series of reactions where our body responds immediately, as well as keeps a memory of this pathogen so that it can respond to that at a much later stage, right? So this is in very simple terms. So basically it produces antibodies against the pathogens. So before the pandemic, there were mainly weakened or inactivated vaccines that were available, and recombinant protein. These are the direct antigens packaged and delivered to the body so that body can produce immune response. And there were also other viral vectors. I will go through them very, very quickly. So the first recorded vaccination was in 1798. I think many of you are familiar with this. In 1798. And then in 1896, we had this killed whole organism. Basically, the dead virus or dead bacterial cells were used as antigens. And then some part of the bacteria was actually used to treat diphtheria and tetanus. And at a later stage in 1970s, when people started working with recombinant DNA technology and able to actually make proteins in large amount, they thought of just using a single protein subunit vaccines. And then with evolution of different technologies, people have actually employed many different methods, right? So, and there are many different ways now we can deliver antigen to the body so that body can actually respond to that. So this is what I'm going to kind of tell you a little bit about today. So before that, so live attenuated virus vaccine. These are the examples of vaccines here. Basically, a virus is passaged through many generations through a cell, so that it actually picks up some mutations which makes them quite harmless. But in terms of their ability to elicit immune response, it more or less remains the same. So this is what is called as an attenuated vaccine. But there are some cases of, when people have used attenuated vaccine. For example, in the case of polio, there was one case of an individual who was vaccinated in 1976. He was found to proliferately shedding viruses in 2005. So this guy had no disease. He was producing a very weakened version of polio virus, but the attenuated strain was not completely attenuated, but it was able to replicate inside the host. So there are those outliers that are always there. And inactivated vaccine, basically they heat kill or they chemically inactivate the virus particles, so it will not have any ability to multiply, but it will have all the antigens that are necessary to produce the immune response. Right? So polio virus and influenza viruses are actually produced by this mechanism. And now, once people started figuring out how to make proteins in large amounts in the lab, they were actually just injecting directly those viral proteins, so that they are given to these the dendritic cells. So a series of events would take place, eventually leading to the elicitation for the production of antibody. Right? So these are some of the examples. Now, once we had a good hold of molecular biology, people started using other viral vectors. You can actually take a piece of, for example, in the case of coronavirus, you can take the spike gene, you can insert into another virus, and when it actually enters the host, you can actually make it produce a large amount of this protein that will then be used as antigen for eliciting the immune response. So this is another mechanism that people have tried out. And there are also these toxoid vaccines, where you basically take the toxins, you heat kill them, but they will not lose their antigenicity. And if you just put them with some kind of adjuvants, they also elicit immune response. Right? And I'll not go through, this is a loaded slide. Most of the vaccines, when they are injected, almost always they're recognized by these dendritic cells, and then a series of things happen. They produce antibodies immediately, as well as they keep the memory of this antigen. Basically, they break up this vaccine, this antigen, and display many different peptides. And so they make a spectrum of antibodies against that particular antigen. Right? And one of the other things that we need to understand here is whenever an individual gets vaccinated, primary response, so this is the threshold antibody level that actually provides protection, it goes down. So the length of incubation is very key. Right? Even if our body has some memory of this particular antigen or virus, if this, some of the viruses, for example, have a very short incubation period. Incubation period is something like from the exposure to virus to the onset of the disease, right? So that is what is incubation period. So if that is really small, then this vaccination is not really effective. Right? So the longer incubation periods are actually much better. In the case of SARS-CoV-2, SARS coronavirus 2, I think it was about 6.4 days. So and for our B cells to start producing antibodies after the secondary, after the exposure to pathogen or the antigen, it takes about 3 days. So by the 4th and 5th day, our antibody levels actually go above this particular threshold. And there are also other, so this is the same thing. If so, for example, Haemophilus influenza has a very short incubation period. So then the vaccination is not very effective. We need to come up with other ways of treating the disease. And there are also other crazy vaccines. For example, you might have heard about yellow fever vaccine, right? And in fact, that is the first vaccine to get a Nobel Prize in 1951, right? So Thomas Tyler, I think his name was, what he did, this yellow fever is actually caused by a family of viruses called Flaviviridae. And those viruses basically infect primates. So he actually isolated a strain of virus from one of the primates, and he passaged them in the primate for several generations, and then took the same and then passaged them in chicken brain, chicken embryo, mouse brain, mouse embryo, finally arrive at a strain that produce lifelong immunity, right? So this is really crazy. We have understood everything there is to understand about this particular antigen, but we still don't know how such a prolonged immunity or protection can be provided. So these are one of the very rare cases. But now people have been studying how soon these antibody titers go down. So now they think that after about 10 years, there may be a need for a second vaccination, right? And if you want to go to many of the African countries, you need to take this yellow fever vaccination. And, Egypt and some such countries. Now coming to mRNA vaccine, I think all of you know this is the holy grail of biology, right? Central dogma. You have DNA, from that RNA is made, and which actually has the code for making the protein. And many of the viruses actually have RNA as their genetic material. I think most of you know about this. So here, instead of taking the whole organism, we will basically look for an antigen, and we will synthesize that RNA and package it in a way that it actually enters the cell, so that our own cells can make these antigens, so that our body can produce a robust immune response, right? So that is the whole idea here. And if you look at the mRNA vaccine history, I have just cataloged some of the major events. I think many of you are familiar with Sidney Brenner. He was the first one to describe what exactly is mRNA. And then the first protein was produced in vitro by using ribosomes isolated from mRNA. And 1978, I think all of you know this company called Moderna, right? Moderna was started by a guy, I think Dr. Robert Langer, who's still there as a faculty at MIT. He founded the company in 2010, and he discovered that when mRNA is actually packaged inside these unilamellar liposomes, they can actually enter the cells. We can transfect human cells. We can make this mRNA enter into the cells, because mRNA is highly hydrophilic in nature. It has a lot of these negative charges, right? And then in 1984, in vitro translation and transcription of mRNA was achieved. Again, these are all techniques, right? And they found a better way to encapsulate this mRNA. And in 1990, this was one of the major breakthroughs. The first this was the first mRNA vaccine, but it kind of faded away because the response was not that great. And from 1990s, a lot of people tried to make vaccine against cancer cells and other things, but they failed, basically due to the issue associated with stability and safety of these mRNA vaccines, right? So there was a period of lull for about 10 to maybe 15 years. And in 2005, working at University of Penn, they made a single change to their protocol by using this modified uridine instead of uridine. They used a pseudo-uridine for making mRNA. So that was the only tweak they made to that protocol, and they got the Nobel Prize for that. And although I think another major discovery came from Robert Langer, but he's a billionaire, so probably he doesn't care about Nobel Prize now. So basically, this is what it is. When you have an unmodified mRNA, which is actually foreign, right? It is coming from outside. Our cells has a way of finding it out, and this mRNA is degraded very quickly. And even you load the cell with many copies of mRNA, it's very inefficient in actually making the protein from them. But as soon as you insert a pseudo-uridine in the place of uridine, basically this magically helps in stabilizing the mRNA. So it stops mRNA degradation completely. right? And probably many of you are aware of pseudoiridine, which is known as the fifth nucleotide. It is quite abundantly found in tRNAs, and it helps in folding and other things. So I really don't know what made them to try pseudoiridine. Probably because it was one of the most abundant modified nucleosides that was there. So they thought of incorporating that. So by 2005, there were other technologies that were developed. So this T7 polymerase is an RNA polymerase. So you can basically make a transcript. RNA, you can synthesize RNA using this enzyme. And that's what they have done. And they have packaged this in a nanoparticle, lipophilic nanoparticle. And they injected, and this is what they have measured. So what they have done is they have measured the innate immune response of some of the cells. And whenever they had all these modified uridine, there was absolutely no immune response. So TNF alpha is basically used as a marker for that. Right? So this is a very elegant experiment and very elegant finding, and they published in a decent journal, Immunity, in 2005. So basically, in a nutshell, this is what happens. You have a lipid nanoparticle carrying this mRNA, which has pseudoiridine, and then it is highly efficiently translated, and protein is made, and protein glycosylation is also a very important feature here. And protein gets glycosylated, and it is displayed, and our immune system basically takes over, right? So these are the B cell receptors, and B cell basically keep the memory of these antigens. So basically there are three ways one can administer mRNA vaccine. Intramuscularly is what is most commonly used, and there are also other ways one can do this. And there are two vaccines, mRNA-based vaccines in the market. One was made by Pfizer, and the other was made by Pfizer also. I think they collaborated with this German company, BioNTech, and Moderna. You see that there is good protection. These are the cumulative COVID incidents after vaccination, and it looks like Moderna performed slightly better. So in terms of stability and the kind of nanoparticle, both of them, their mechanism of action, they are very similar, but there are some slight differences in their composition. So if you look at the traditional vaccine development pipeline, it used to take about anywhere from 10 to 15 years. Now in this case, especially, it took only 10 months, right? So in 2019 December, the SARS coronavirus sequence was published, and within first 10 months, and because the mRNA technology was also ripe, they were able to make the vaccine and get all the approvals in a record period of time. And I think rest is history, as all of you know. So this is what it is. So it took about 10 months. Compared to malaria, so we don't really have a robust vaccine against malaria, tuberculosis, even dengue virus and all. But in most other cases where we actually have vaccine, it took a lot of time. For example, Ebola virus, 1976 to 2019. Even for HIV, we still don't have any vaccine. But the first case was not in 1983, it was way back in, I think, 1956 or so. So the technology to just produce the antigen or the mRNA coding for antigen in the lab, and even I can produce tons and tons of these mRNA vaccine in the lab, right? And that is the beauty of this. As against if I want to produce live or attenuated vaccine, I have to have BSL-3, I have to culture the cell lines, I have to infect, I have to make large amount of virus, then inactivate them. It's a long and drawn protocol, right? Whereas this actually cuts the cost down. And now, even if you have any new disease, any new virus, within 2 months, one can make a vaccine for that. So something some people might be interested in, COVID-19 vaccine safety. Again, some of these data all I got from CDC. So anaphylaxis is something like an allergic reaction against the vaccine. 5 cases per million doses, and report of death. There are all kinds of data out there. I really don't know which one to believe, which one to not, because how do you associate vaccine as the cause for death, especially during COVID, right? Not very sure. And GBS syndrome, they are very rare. Some cases, the immune response that is generated against the vaccine can damage the nervous system, right? In some very rare cases, and that will paralyze that individual. And myocarditis, probably some of you know about this. This is inflammation of the heart muscle, right? This is again, these numbers are, I really don't know, because CDC has not reported them, and I've taken them from what I think as reliable sources. Again, these are not really direct. There is no direct correlation between with vaccination, but these are the conditions that were observed after vaccination, right? Thrombosis, blood clotting, and other issues. So this is what it is. And if you look at, again, this I've taken from CDC, and if you look at vaccines life cycle, from basic research to post-approval, safety is a major priority, right? And so if you have any questions, I'm free. I'm understanding.